Hey everyone, in this video I'm going to be discussing the paper Action Minimization Meets Generative Modeling, Efficient Transition Path Sampling with the OM Functional. Okay, so this title seems super complicated and I agree, it's super daunting, but this paper was just, recently submitted to the ICML conference in Vancouver, ICML 2025, and it's really interesting and how they propose a kind of different way to do this to do sampling such that when we want to observe, states in between two given states. So say I want to observe the path from a protein going from one configuration to another configuration. This paper was kind of saying, how do we understand what the path between those two configurations are and how can I sample along that path? And it specifically focuses on how we can use pre-trained generative models. So pre-trained diffusion models. So there's a ton of big models out there that have been trained on huge corpuses of data, right? And so we want to be able to use these pre-trained models and how they can enable efficient molecular transition path sampling using what internally in generative models are called score functions. And these score functions, we use them as kind of pseudo force fields that tell our configuration how to move in like molecular dynamics type terms. Sounds super complicated. I promise it'll get hopefully a little clearer through the course of this video. This will be a little bit more technically challenging than the last video, but I think it's still super interesting. And yeah, let's get into it. Okay, so what is transition path sampling? I know I mentioned this, but the goal is to kind of find likely meaning high probability paths a molecule can take between transitioning between two stable states. So this is kind of like if we have a reaction, we have a transition state in the reaction, we have our two stable states, which are like the reactants and the products, right? And we have a transition state. And this is kind of that's exactly what we're doing, transition path sampling. But it can, it doesn't have to be a reaction, it can be two configurations of a single, molecule. Like, so for example, in proteins, so it's important for understanding chemical reactions, protein folding, and biomolecular changes. So for example, protein going from one stable state to another stable state and changing its configurations. And these kinds of transformations are really important to understand, right? So you can understand how can you maybe stabilize a transition state, or how can you bind something to a transition state of a configuration of a protein or something like that, right? But the challenge is, what is the challenge? These transitions are rare. You have to overcome an activation energy barrier. And, as from all these sampling videos, when you have a high energy activation, very difficult to sample. Right? It takes a long time, requires long simulations, or very smart collective variable design. This is the same kind of idea as reaction coordinates. And it's very computationally expensive, right? So we want to be able to solve these problems. What was the previous work? Let's talk about that first. So we have the traditional approaches, right? We've talked about this before, metadynamics, umbrella sampling. These all require kind of predefining our collective variables or our reaction coordinates. You can use these terms I'm kind of using interchangeably, but it's the same kind of idea where you need to have some one-dimensional coordinate that you're sampling along. Some other work that's been done is with atomistic generative models. But usually these generative models, what they do is they produce unbiased, independent samples from the set of configuration of molecular ensembles. So usually it's just kind of producing for example, say we're looking at protein configurations, it's just kind of producing a likely configuration of a protein. And we can use this to adapt it to perform rare event sampling, but it's a little bit difficult. What this paper in particular tries to do is do this kind of thing called generative model interpolation. And the idea is, we talked about when we talked about VAEs in the RAVE paper, there's this idea of a latent space in these generative models. And instead of doing interpolation is basically like connecting the dots, right? Instead of connecting the dots in the in the space of configurations, which is the direct XYZ Cartesian coordinates, we can connect the dots in the latent space of the generative models and then kind of map how we connected the dots back to the real space. And there's some benefits in doing this. So for example, say we, just try and connect the dots between configuration A of a protein and configuration B of a protein, right? And we just do this in the Cartesian space. So connecting the dots, I literally mean, we have the X coordinate, XYZ coordinates of one atom in configuration A, XYZ coordinates of one atom in configuration B. And we're literally say linearly interpolating it. And we're saying that's how the motion of this atom is going to work over from the transition state from A to B. This can lead to super wonky, configurations in the middle, right? That are totally physically unreasonable, especially if we're doing this because we're doing this for every single atom in in the protein. So instead, what we can do is try doing this sort of connecting the dots, but within the latent space of generative models. And the idea is the generative models in this latent space are going to have learned what is physically reasonable by training on, only valid configurations of a protein. So when it learns what's physically reasonable, hopefully in this latent space, we're not going to get super wonky configurations. And this has been done a little bit in the past, but this paper kind of takes a unique approach to how this interpolation is done. So let me talk a little bit about what generative models are. Okay, so generative models, and this paper focuses on diffusion or flow matching models. They learn the distribution of valid molecular configurations from data. So just training on a huge, set of protein configurations, and it's kind of learning what is valid, what is not. Typically, it's used to generate independent static samples. So it's used to generate, what is a configuration of a protein, just kind of generate one static sample. And this kind of class of models, right, includes VAEs, which we discussed in the RAVE paper as well, but also diffusion models, which I'll talk about more in this presentation. And the idea is, in this paper, we want to understand, can we use these generative models to instead generate paths, right? Sequences of configurations showing realistic molecular transitions, not just independent static samples. Okay, so these are just some types of generative models. We have GANs, which I'm not going to focus on in this discussion at all, but they're another interesting type of model. We've already talked a little bit about VAEs previously. There's kind of this idea of an encoder, right? We learn an encoder to get from an X to a Z, and this Z is in your latent space. So I'm going to talk a lot about, just in general, I'm going to mention latent space a lot, and this is just the space within where our Z's live in, right? So we're learning some way to encode X to Z's, X to our latent space. And we also learn a decoder at the same time, right? We're learning some way to get from Z back to X. So in diffusion models, our encoder, it's kind of a little bit different. You can see it at the very bottom, right? I'm also not going to talk too much about flow-based models, but this paper does show how it can work with flow-based models as well. Flow-based models, your encoder is this flow, right? And you're learning kind of an inverse flow as well. In diffusion models, right, we have a kind of interesting setup. Our encoder is kind of a block that keeps adding Gaussian noise to our input. So we take our input, and you can see these right arrows. We're kind of just making it more and more and more noisy. We're gradually adding Gaussian noise. And by Gaussian noise, I just mean like this random normal noise. So we keep adding that, and then we finally get to Z, right? And our decoder is what we kind of learn that takes this random seed Z in our latent space and denoises it until we get back to kind of our X space, which is the configuration now. This X, whatever we generate from this denoising is the configuration of, say we're doing molecular design, protein design, the configuration that we generated. So that's kind of the idea, is we always have this kind of encoder, decoder setup, I guess, but how exactly they're set up is a little bit different. So now that we have the general idea of what these diffusion models are, let's discuss what the score functions are. Okay, so this is a little background on what is the score function. This is the specific equation for the score function. Score function of a specific, at a specific x point, is equal to the gradient with respect to x log p of x. P of x is the probability density at that configuration x. This equation, seems complicated, but the basic intuition is we're just kind of, the score function is pointing towards, right? It's a gradient, so we're getting a vector. We're getting a vector that's pointing towards directions of higher probability, right? Because we have a gradient of log p of x, so we have something that's kind of proportional to the probability p of x. So the score function is pointing towards directions of higher probability, kind of like a compass that's guiding us from unlikely to likely molecular structures based on how this generative model has learned. And the idea is these generative models learn this score function while training, okay? And what this paper kind of is proposing is that this score function really can act as a force field, like a field of forces learned from data, instead of something that's actually run with physics, right, through molecular dynamics, where we actually compute all these forces using some interactions and Leonard-Jones potentials, all this sort of stuff. We can learn like a sort of force field through these score functions, showing how to nudge these molecules towards more valid configurations, right? More energetically favorable or more probabilistically favorable conformations. So that's the kind of underlying idea of how this paper proposes using score functions. Now I'm going to go into a little bit, go and discuss stochastic dynamics. And I promise we're going to tie all of these together, but I just want to make sure all these key parts are understood before. So stochastic dynamics is just a way to describe, motion, for example, in general, where we have, some term that's called a drift term. And this drift term is what's actually, for example, the forces behind the motion. But we also have like random motion, right? At any temperature, at any non-zero temperature, we're not just going to have completely deterministic actions. We're going to have random fluctuations that come just from the energy of the temperature of the system. So the idea is we want to be able to describe both this random motion, the combination of the random motion, and the drift, or the kind of force energetically favorable motions. So just imagine molecular configurations moving in random noisy conditions. So we have our Brownian motion, which is the random motion term, and the drift, which is the force field. And so that's what this SDE, or stochastic differential equation, is basically saying. dx is the change in our actual configuration, is going to be equal to our score function, dt. So we have our force, right, our acceleration, basically, dt. And then we have... or velocity dt, something that's describing how this motion is going to happen. And then we have our random, what we call wt, dwt, which is just our random Gaussian noise that's kind of added to this term. So that's basically what these bullets are explaining. And the interpretation is, this kind of describes how this generative model is going to induce random walks guided by probability directions on the molecular shape space. And finally, now I'm going to introduce the OM action functional. This also seems super complicated. I promise it'll get a little bit more clear once I explain this. This functional basically says, given a stochastic differential equation, so given the differential equation I proposed to you on the last slide, what is the path of maximum probability, like from point A to B, right? And so the idea is that the probability of x, this x is a path, actually. That's why we have this x parentheses dot. That's saying like across time, x is a function of t almost. So the probability of this path is proportional to e to the minus s of x of this path. And so in order to maximize the probability of this path, what we want to do is minimize this s of x, right? Because proportional to e to the negative s of x. And s of x is equal to 1 over 2d, this term here, right? So d is a hyperparameter, but we basically have this a, b, and c, these three terms. So the idea is to minimize s, we want to minimize a, b, and c. And so this, like how we get this, that's a long derivation that they show in the appendix of the paper. But this is also kind of like a known thing. This OM action functional is a known way of figuring out the path of maximum probability given like a force field, I guess. And so that's kind of the key idea. We want to minimize A, B, and C. So let's look at all of these terms, and I'll just kind of explain it intuitively. This A term, so we can see it has something to do with x at I plus 1, the difference of xi plus 1 and xi. So the norm of this difference squared. And the idea is this is basically a smoothness term. We want to favor paths, remember we want to minimize A, so we want to favor paths generally when x doesn't change too much from one time step to the next. We also can see there's this delta t on the bottom here, right? So if we have very small time steps, we really want x to not change much. But if we have larger time steps, x can change a little bit faster, right? And this makes sense. If we have small time steps, our configuration shouldn't change hugely from one tiny time step to the next, right? It doesn't make sense for that to happen. It's not physically reasonable. The B term says something about phi of xi, and this phi of xi is basically the force vector or force field. We can think about it like that. So we call this the drift norm term, and the idea is to penalize paths with high vector field strength or high, magnitude forces, because we want to go on a path with forces that are, generally physically reasonable. If we end up in these weird corner cases where the forces are huge, it doesn't make much physical sense. So we want to go on a path with physically reasonable forces. And lastly, we have the C term, which is the divergence term. And the idea here is we have a divergence of phi of x, right? So this measures, I guess, how much force field emanates from a certain x point. So the idea is if we want to minimize this divergence, we want to encourage paths to travel in convex parts of the force landscape. We want to encourage basically staying at near or low energy points. And that's basically the idea of these three terms, is they're all very mappable to, I guess, physically reasonable explanations, right? We want things to just be physically reasonable, and these three terms kind of explain that. So where are we now, right? There's been a lot of math, there's been a lot of background. Let's summarize what we know and how we can try and put these tools together. So this is a direct quote from the paper, but I thought it was a great way of summarizing where we're at. To use this OM action to compute path probabilities with the DDPM. DDPM is the generative model, the diffusion model. We must construct a surrogate stochastic differential equation such that paths under this SDE have high likelihood under the data distribution used to train the model. Okay, that's a lot of words, but I think it really well summarizes where we're at. This DDPM, this diffusion model, gives us some score function. Given a properly formed SDE, we can find high likelihood paths using the OM action, right? So the OM action is a tool that gives us the paths. The question we need to answer is, what is the best way to construct an SDE, right? I've been constantly talking about, oh, let's use this score function as kind of like a pseudo accelerate, pseudo force, right? But how do we directly kind of construct that SDE? How do I map that from the generative model idea back to the physics, right? Such that these high likelihood paths in the SDE are equal or equivalent to high likelihood paths under the true data distribution, i.e. high likelihood paths within the true space of configurations of a protein. So let's talk about how we can construct that SDE. Again, as I mentioned many times, this is coming from the idea of using this learned score function as a force field. And we want this SDE to look like exactly what I had previously, right? We have this score function dt plus a random process, right? Score function dt gives us a drift, random process gives us the random noise. And we can think of this SDE as the one that arises from iterative noising and denoising. So what we do is we say, okay, from x0 to x1, right, we noise to go from x0 to x1, right? We add some noise. This is like our random, I guess, kind of random process. And our denoising step is where the score function comes into play. The score function comes into play during our denoising in a generative model in general. And so that's how we kind of get both of these terms put together. We have our randomness coming from the noise part of it, the noising step. And then we have our score function, which is kind of the smart denoising by pushing things towards valid configurations. And that comes from our denoising step. So that's basically what I've written down. The score function push comes from denoising, random additions come from noising. And that's just the key kind of bridge between how can we take what happens in the generative model and kind of map that to an SDE that happens with our kind of physics brain intuition. Okay, so putting everything together. The score function from this diffusion model gives us a force field. And from this, we can construct the stochastic differential equation easily, right? Through this noising and denoising process. The most likely path is then given by minimizing the OM functional. The key assumption we are assuming, though, is that the score function can truly be interpreted as a force field, right? But the score function nudges these configurations to those of higher probabilities. What does higher probabilities mean to the model? The probabilities are based on the data the model was trained on, not actual true physics, right? It's based on the corpus of protein configurations that it was actually trained on. So this interpretation really holds if what we trained on was Boltzmann distributed. So if higher energy configurations were much less likely to be in the corpus of data versus low energy configurations, right? If that proportion was correct in the trained set, then this interpretation would hold. And then we can treat the score function at x at tau equals 0 as an atomistic force field. So this is an important point. The last bullet, the generative model is actually learning different score functions depending on what quote unquote level of noise we are at. So let me go back to the previous slide just to show you what this means. In this image, right, we have all these different x0 to x1 to x2 to x3, all the way blah, 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 blah to z, right? But each of these x0 to x1, for example, is a noise step. And if I'm trying to map back from x1 to x0, we're going to have some score function that comes into play that nudges from x1 to x0, nudges intelligently, right? But the nudge from x1 to x0 will be different than the nudge learned from x2 to x1 and x3 to x2 all the way down, right? So these score functions are different. They're based on some hyperparameter, tau. And tau is telling you what level of the noise are we at. So this level of noise is the tau parameter, and the true final force field will be given when there is no noise in the space we are looking at. I.e., we are looking at the space of configurations. We are just looking at the space of configurations, right? So this tau equals 0 means that there's no noise in the space we are looking at. So now we have, like, I guess, the true force field. But we could just look at the score function at any value at tau as the force field at that specific noise level, right? This tau doesn't really matter. And then we could encode our starting and ending states to that noise level, right? And construct our path in the noisy latent space. So if we have our score function at a specific certain noise level, we can map our start A and B points and just do our OM functional, math in our noisy space, in our tau, is non-zero tau space, and then just map all those paths, decode them, I guess, back to valid configurations. So tau is really just some hyperparameter that we're allowed to tune. So let me just put everything together and show you the algorithm they came up with. First, this is basically the general algorithm they came up with. Find this optimal parameter tau, optimal hyperparameter. Then we just initialize the path as some guess in some latent space. So what we do is compute some initial guess, right? And this path basically is some kind of interpolation. They show many types of different ways to do this sort of initialization. It doesn't really matter, right? But then the idea is we keep updating this path to gradually minimize the OM action of this space. Or we're trying to basically, if we're minimizing the OM action, we're trying to maximize the likelihood of this path under the score function at this time or noise level tau, right? So if this path is constructed in a noisy space, we get this path at the end of this algorithm, and then we just denoise the values on our path, or decode, or whatever you want to say, until we get our path in the true configuration space. And that's the whole algorithm. It was a lot of complex, I guess, background and math, but the algorithm is super simple. We're just initializing some path in some latent space of this diffusion model, and just keeping on updating this path using some form where we were trying to minimize along this path some value, right? We're really just doing a path minimization problem where we're trying to minimize this OM action of this path. And we do this just by following simple gradient descent. We compute the OM action, and then we do a little update of our path. And then we compute OM action and do an update, and just keep doing that iteratively until we've converged. Okay, so here's one, I guess, I think, great image that they had in the paper that kind of just summarizes one of their results. They had a lot of different results, and they tested this on many different systems. But this picture here, we can see in picture A, we see the free energy profile of 3 different protein folding processes. And overlaid on these free energy profiles on the right side, you can see the OM sampled transition paths. And it looks actually pretty good, right? You can see stuff going from one point to another point of the free energy landscape, and it looks pretty smooth. And in image B, we can see for the specific BBA protein folding, so that's the image on the top right of image A, we can see along those paths that were sampled, we can see 4 different transition states that were sampled on the 4 different sampled paths. These transition states look pretty reasonable. They don't look crazy, which is good. And images C and D kind of just compare this method over traditional molecular dynamics so over MD. You can see that the sampling time, right? So just the time it takes for this to run is orders of magnitude, just much, much lower than traditional molecular dynamics, where you have to truly compute all these forces and do everything at every step, right? Whereas here, you're not really computing any forces. You're just taking a pre-trained model that already knows the score function, The model already has this score function within it. So you don't even have to, you're not trying to train the model. You're not trying to do anything. You're trying to take these models and just use the information it already has as your force field. That's why the computational time is so much lower. And for proteins we actually know the processes of, it does pretty well. So you can see this Shannon divergence. So these orange and pink things on the bar graph is the OM method that they've been discussing, and the blues are the MD simulations. And you can see, like, for this Jensen-Shannon divergence, you want lower values. That's better, right? Better, more close to reality. You can see the OM models do very well. For the valid transition path rate, you want higher values. Again, do very well. And you can see just the results show very promising results compared to much longer running, right? Much longer time taking molecular dynamics simulations. Okay, now to just wrap this all up, what's the conclusion? The idea is we want to repurpose, what this paper did is repurposing pre-trained generative models for efficient transition path sampling. We don't need to train a new model. We don't need to compute forces. We don't need to do all this. Super computationally efficient. It interprets the model's learned score function as a drift term in stochastic dynamics. And OM action minimization finds physically realistic paths by optimizing the movements along this force field. And overall, this method is scalable and flexible, right? It works with existing generative models and easily adapts to new systems. In the paper, they also went into showing how, they were able to generalize using a model that was not really trained on a given protein at all. They were able to take that model and find the transition paths of a completely, different protein, right? And so that just shows that these generative models don't need to be trained on your specific, use case, which is much more difficult. Like, if we had something that was trained on our specific use case, it would be kind of useless because we wouldn't need to use it to learn about this specific use case, right? Because we would already have had that training done. So the idea is it's super generalizable. We can take something that was trained on, a different selection of proteins and apply it to a new protein. It easily adapts to new systems, and it's very efficient. They show that it's orders of magnitude fewer evaluations than traditional enhanced sampling or molecular dynamics. And so I think this paper is super promising, and we'll see. Hopefully it does well, and hopefully people use it. All right, thank you guys so much for watching, and I'll see you in the next one.